hopefully hopefully you're here to see Bryan, because that's what's going to happen here. Bryan, go ahead and defend yourself.
All right. Okay. So everyone in this room started as one cell. So you started as one cell, you started as one cell, okay? But that cell divided, and it divided again, and it did this many, many times, and eventually you're about 10 trillion cells today. That's Richard Feynman, if you guys didn't notice. Okay, not only do you need cell divisions to get where you are today, but just to stay alive, you need about 10 billion cell divisions every day. So right now, while you're sitting here, you got a bunch of cells dividing. Okay, so what happens when a cell divides? So this cell has got to make sure that half the DNA goes to each daughter cell. Because if the daughter cell doesn't get the DNA, it's going to be really messed up, right? DNA is important for the cell's function. Can everyone hear me okay? Mike's good? Okay, great. Yeah. Okay. Up? All right, sorry. Is that better? Okay. Okay, so what makes sure that half the DNA goes to each daughter cell? Well, there's this thing called the spindle. And what it does is it grabs the DNA and it sorts it and does all this stuff, and it ensures that each daughter cell will get half. So what we're going to see here is a cell where DNA is in red and the spindle basically is going to be in green. And we're going to see it kind of grab the DNA and do its thing. So let's watch this. Oh, right? And then, oh, see, divided it. All right, so, right, it's really important. So besides being like a basic biological process that's really interesting, it does have medical applications. So if you have errors in this segregation of DNA early in development, you get birth defects. So like Down syndrome is one of the things that can happen from errors in segregating DNA. Furthermore, the most prominent chemotherapy is a drug called Taxol. And Taxol works by disrupting the spindle. So by disrupting the spindle, they stop cells from dividing. And so if we understand how the spindle works, in theory, we could develop better chemotherapy drugs and maybe understand cancer better in general. But that's only one reason why I study it. I think the real reason I study it, I think spindles are really, really cool. And yeah, so this is like the coolest thing. So like from an engineering perspective, kind of like this thing is just like, it's like all these awesome properties. So like what happens when two spindles touch each other? Is it going to be like time cop style where it implodes? Or like, let's see what happens. Okay, no one's seen time cop, it's okay. All right, so you see that it merges into one like super spindle or something, right? So that's really cool. You can also like ablate a spindle. So here you took a laser, we cut it, and let's see what happens. What happens when you put a big hole in this spindle or a line? You get this wave, but then it reheals. So it has a lot of other cool properties, but I just wanted to show a couple of videos just to motivate why it's such a cool thing to study. So I've been talking about it, but what is it made of? So the spindle is made up of a bunch of tubes, and the tubes are really, really skinny. So they're called, or they're really small, so they're called microtubules because they're small tubes. They're about 25 nanometers across, and in the spindle, they're usually about 6 microns is their average length. This spindle is made up of a protein called tubulin. This protein is really a heterodimer between beta tubulin and alpha tubulin, but this is such a strong bond that this is our basic subunit, and we're just going to call that tubulin. So for the rest of the talk, when you see this, just think of like one thing, tubulin. Okay. So here's an idea. I want you guys just to be familiar with this idea of like monomer and polymer. So mono means one, so these are like one things. Poly means many, so these are many things. So you can have monomers around, and they can form into a polymer. And so like in this cell, just to give a picture, you really have these monomers kind of diffusing around, and they're kind of growing these polymers. So it might look something a little like this. Just to show one more time, not everyone works with cells and stuff and has this picture, but just the idea that there's stuff around that can randomly do that. It took a long time to make in PowerPoint. All right, so now we have our basic understanding of monomer and polymer. Let's see, okay, so spindles are made of microtubules, and we're going to try to understand microtubules to understand the spindle. So microtubules can really do 3 things. The first thing they can do is they can nucleate. So you have to go from these kind of monomers to your first kind of like stable polymer segment, where you start getting those growth I was showing before. Once you kind of get this, and in cells I should say that this nucleation is really facilitated and heavily regulated by a nucleator. And it just happens to be that way in cells, that you need this nucleator to do this. And this is just a separate protein in the cell. Okay, once you have them, they can grow and shrink. And so here's a cool video. So what we do here is we take this first part, and it's kind of like chemically stabilized. And then you're going to look at these monomer guys, and they're like labeled in green. So we're going to see these green monomers are labeled in green grow. And we'll watch it grow and shrink. So let's see. So the pink things are now stabilized, but we see these microtubules grow, boom, shrink. It's kind of cool, right? Okay, so they can nucleate. Once they're there, they can grow and shrink. And then finally, they can also interact with each other. So there's a lot of other accessory proteins that take these two microtubules and slide them across each other and bind them and bundle them and all kinds of things. So here in red is one species of microtubule, and green's another. And we're going to see them kind of run along each other via these accessory proteins. Okay, so now we've seen the three things that they can do. Let's look at like a real cell. So here's a frog egg, and it's about 1.5 millimeters, and microtubules are labeled in red. So you see like, okay, there's something there. Let's go see what that is. Let's zoom in. Oh, it's our friend the spindle, right? So in green is DNA. And so, okay, so now we see a bunch of microtubules here and not there. So why is that? So the cell has to do something, right? We know the cell can affect basically three things. It can affect its interactions, it can affect its growing and shrinking, right? Or it can affect its nucleation. And it's got to affect that in some spatial way in order to get stuff here, but not there. So naively, from what I've told you, let's like start testing a few of these hypotheses, right? Maybe you're like, the nucleation's the same everywhere, the growing, the shrinking is the same everywhere, but for some crazy reason, as soon as a microtubule is created, it gets transported into this area. It's kind of a crazy idea, but you know, one way to rule it out is a lot of really smart biologists and biophysicists have measured how fast microtubules can slide and move relative to each other, and it's just not possible to move them there that fast. So we can rule out kind of interactions. The next good question, or I think the next good hypothesis would be how long a microtubule lasts for could be spatially regulated. So maybe you nucleate the same everywhere, but right around DNA, you've like selectively stabilized them. And that's why you have a lot of microtubules. And that's a good hypothesis, except except then Dan during his postdoc did a lot of single molecule speckle microscopy measuring the lifetime of these microtubules. And what he found was the lifetime in the spindle is the same as out of the spindle. And furthermore, even within the spindle, the lifetime does not change. So it's not selectively stabilized. So the last thing it has to be is nucleation. You know if the lifetime's not changing, you know it's not some crazy interaction model. Basically, it's the nucleation that's happening at DNA or localized to DNA that gives us a spindle there. And that's what this says. Okay, so let's go take a look at that same egg. So that egg came from this frog. You inject this frog with a hormone, drops a bunch of eggs, you spin it, and you spin it really fast. And then in this area here, it's called a fraction. This fraction corresponds to the inside of this egg. So the cytoplasm of that egg. And so the reason I do this is it's really easy for me to add and deplete stuff from it. And furthermore, it gives me like bigger volumes to deal with. It's just a really nice model system. Happened to be pioneered by like Andrew Murray and Tim Mitchinson, just for a little Harvard history, I guess. Okay, so why do I like to use it? Well, one thing that's great is if you add DNA and put it through a cell cycle, you can get a spindle to assemble. Here's a spindle assembling just from adding DNA back into this. So like that inside of the cell was ready for DNA to start, you know, giving you nucleation and thus a spindle. All right, so now we take that same extract system, and I was saying DNA triggers this nucleation. Okay, so it triggers it through like a cascade of proteins. You don't have to add the DNA. You can add one of these signaling proteins lower, and you still get this thing, right? If you have this pathway, you don't have to start at the top. You can start a little bit lower. You still get this stuff. So in this case, the protein is called RAN. Constitutively active form is RAN-Q69L. So if you add this, you're just going to activate nucleation everywhere. And that's what Sabina does in this very beautiful movie. And so what they're going to do is they're going to have tubulin in red. So all those monomer guys are in red. And then if you see red lines, it's presumably a microtubule or a bundle of microtubules. And then in green, they label a protein called EB1, which tracks growing ends. So you'll kind of see the growing end of a microtubule. So you can kind of see growing ends in green and then try to see stuff in red. And what we hope, or what they're going to see is it looks like microtubules are growing along each other and then possibly branching. And so let's go see that. Oh, I guess I could have pressed play on this thing. Sorry. OK. So you see them start to like, possibly these are nucleated along each other, right? Pretty cool. OK. So from this and other data as well, there are kind of two extreme hypotheses you can imagine, right? So one is because you saw microtubules kind of like go along each other, you might say, well, maybe that microtubule OK, so first, presumably this nucleator is activated at the chromosomes. But maybe it's further regulated by microtubules. So maybe after it's activated, it needs to bind this microtubule, and then it starts growing. That might be one explanation for the video we saw. That's called the bound nucleation hypothesis. So we'll note that these two nucleators up here that are not bound, they're not going to grow anything. But this guy, once he kind of binds, now he can nucleate. And what we call the all nucleation hypothesis, you don't care if you're bound or not. Whether you're bound does not affect your nucleation activity. So in this case, you'll notice these two nucleators that don't bind will still nucleate. And still, by diffusion, you could be bound, but basically your activity doesn't care about your localization. So these are two extreme cases, and we kind of want to investigate this. So, OK, so it's hard to use imaging to study this because in the spindle, these guys, like there's such a high density of microtubules that like direct visualization of microtubules and branch things can be difficult. And then furthermore, if you kind of look at this picture, to me, they don't look so different, right? Here's a branching here's a branch, and here's a branch. Now, yeah, you do have a guy out here, but maybe something could start nucleating and unbind, then diffuse off. So I think even if I could get an image somehow, it would still be challenging to really rule out one hypothesis. So we decided instead to look in the spindle is to kind of look outside the spindle. So if the bound nucleation hypothesis were true, remember, we're activating nucleators at this chromosomes. They're binding and unbinding microtubules and diffusing. We expect some to diffuse away from the spindle boundary. Now, since they need to be under this hypothesis, if it were true, they would not nucleate as they leave because they're not bound to anything. And we'd expect the polymer as a function of distance away from the boundary to basically just go to 0 very fast. However, in the other case, where nucleators are active when they're not bound, then when they leave the spindle, they continue to nucleate. So in this case, we expect polymer to continue growing as you leave. And if you do some back-of-the-envelope calculations, you expect the length scale of polymer as you leave the spindle to be 10 microns or possibly more. OK, so this is kind of our starting point, right? We have these two hypotheses. We have a way to rule out them, right? We can check the length scales. If they don't match, we can kind of cross it off. All we have to do now is just measure the polymer concentration. Yes? OK, that's a very good question. So in this cartoon, OK, so in the real spindle, it's not an like infinitely sharp boundary, which is a very good point. So that's why we think, so what we do in practice is we do an intensity threshold, and that gives us a rough boundary. But I think the real boundary is a bit soft, and it could be 2 microns or so. Yeah.
Would you expect also there to be a gradient of active RAM?
Yeah. So. So interestingly, when you look at the distance you expect active RAN to be, I think this is right, from Doogie's thing, it's actually very, very, very short. That's correct, right? Yeah, OK. I just want to make sure I wasn't confusing with another person's paper. So from Doogie's measurements, the actual, yeah. As well? OK. But I remember Doogie's. I just want to make sure I wasn't confusing it. Maybe I was thinking someone else's. So anyways, it's a very, very short gradient. So as it is, in the spindle, I think there's the RAN gradient itself isn't as important. Oh, it's obviously very important, but it's the further down, the pathway further down the biochemistry. And so that basically goes to the edge of the spindle. Like those nucleators, so I'll give you like a good argument to say that a nucleator has to be able to make it to the edge. Maybe that's not the question, though. I'm sorry. That's it? OK.
Well, can I follow up?
Yes.
So just a part nucleators.
Yes.
Just thinking about the microtubules themselves.
Yes.
What is kind of the typical width of the interface, width of the boundary there? Is it greater than 1 micron? Is it greater than 10 microns? Is it
Yeah.
less than that?
So if you just like look at a spindle, what you'll see is just by visualization, if you just look at like an intensity plot, let's say, right, and just like look at an image, it looks about 2 to 3 microns. Yeah, I would say 2 to 3 microns. And it could be from the fact that, near the edge you have guys either branching or you have interactions, or microtubules themselves can diffuse, some length. Also, you take images, maybe like stuff moves a little bit during your imaging. So all these things, I think, gives you about 2 microns.
So that 1 micron would be sort of...
Yeah, this would be like, this is like a theoretical, like this is sort of like order 1 micron. I really am expecting 2 to 3.
Right, but you're really only going to be able to tell if you see this, something much larger than 2 to 3 microns, whatever the interfacial width is, which of these mechanisms.
Yes.
Working.
Yeah. But yeah, the spindle boundary is a good question. So it's not like an ideal case you want an infinitely sharp one, but in reality we have one that isn't quite as sharp. But there is kind of this implicit assumption that at some point there's no more like branching of stuff outward, and there's some amount of diffusion. So it is true that if I were to measure something, well, I haven't shown the data, but let's say I measure something like this, I'm going to have to check that if I see a bunch of polymer out here, that these aren't secretly connected to each other and kind of like nucleating off each other. I'd have to check that. But we'll see what the data is first and see what we have to check. Very good questions. Okay, but this isn't really, so this is like the polymer picture, but the real picture is probably something closer to this, right? We have monomer diffusing everywhere. So this makes it a little more difficult to try to measure the polymer. Furthermore, because the spindle is like actively polymerizing, that could deplete the monomer in that region, which would give you a gradient. So it might even be this. I mean, this is a plausible monomer gradient. It's hard to say what the monomer is really doing. But because we have this complicated picture, it makes it difficult, right? Because I want to look at a position and I want to be like, how much polymer do I have there? But polymer and monomer are made of the same thing. So like, how am I going to know, like, if I just know how much stuff is there, how much tubulin is there, that's not so helpful. I need to know the state of that tubulin. Is it in a polymer or not? And that's a very, very like nanoscopic question, right? So we kind of turn to a length scale that we think makes sense, which is like on that same length scale. So a short length scale measurement. Let's get a quick drink of water. Okay, so there's a process called fret. And what how fret works is you excite a donor fluorophore. And once it's excited, it could pass its energy to an acceptor. However, this can only happen within 5 nanometers. So we're going to use this process, and what we're going to do is we're going to label some of our tubulins with donor, some with acceptor, and we're going to hope for a system like this. I haven't shown you this is how it actually works, but this is what our hope is. When tubulin is just in the cytoplasm, it's just going to freely diffuse, and it, on average, is too far to fret. But what's something that brings tubulin together? Well, a microtubule, right? So maybe, just maybe, a donor will be close enough to an acceptor to fret. And so the idea here is if you need microtubules to fret, then the more microtubules you have, the more fret you'll have. So this is like our idea of our readout for polymer. Okay, but fret is like this physical process. I haven't told you how I'm going to try to measure fret. I'm going to measure it with something called phlim. So here's how phlim works. You send in like you send in some light, and it excites your donor to an excited state. Then that donor can relax and emit a photon. It could also by relax I mean go to it absorbs energy and has to get rid of that energy. So it can get rid of it by emitting a photon. It can like shake it off as heat. And if you have a donor that does that, it generally is in this excited state for about 4 nanoseconds. However, if I add another pathway of relaxation, in this case fret, it's another way that donor can give up the energy, I now have a short lifetime. I just have one more way of relaxing. And in practice, this is about 1 nanosecond. Okay, so now the question is, if I can measure these 2 lifetimes, I can know how many of the donors are in a long lifetime. If I know how many are short, I can get how many are fretting and not. So let's see what happens. So I take some tubulin with donor and acceptor. I don't add any Taxol. Presumably they're all in monomeric or something like this form. And we see one time scale, 4 nanoseconds. Okay, great. That corresponds to donors not fretting. Now if I add Taxol, we see the appearance of this short lifetime. So this argues that these donors are engaged in fret. Okay, so now the question is, I need to say what part of this curve, how much of this curve is 1 nanosecond and how much is 4 nanoseconds, because that'll tell me how much corresponds to guys that can fret and how much can't. And if I know the fraction of guys who can fret, I can infer the fraction of tubulin and polymer. And if you're interested, and then this you can kind of see by eye, but in practice, what we do is we do some like fancy Bayesian analysis to say like, okay, these two amplitudes are consistent with the data, but these two are also kind of consistent, and try to map out the probabilities of different amplitudes of these length scales. If you're interested, Peter and Tay and I and Dan have a paper out that describes it in more detail. So what I want you guys to remember from here is the shape of the curve will give you the fret fraction, but the area under the curve will give you the intensity. So just from the shape, I get what fraction of things are fretting, and then, again, the area under the curve's intensity. So this is kind of nice because it sets me up for the measurement I want. Fret gives me the fraction of tubulin and polymer. This equation just says the fraction of tubulin and polymer, and then there's a parameter for saying that not all donors in polymer can fret. Intensity is great because it gives me like the total amount of tubulin. So I get brightness from monomer, from polymer that can't fret, and from polymer that frets. So this is setting me up well, right? If I have a location and I want to say what's the polymer concentration there, I just need to know the total amount of tubulin there and know what fraction of that tubulin is in polymer. Then I know what the polymer concentration is. So this is all great and dandy and everything, but the problem is it's really hard to know if this is a good description of my fret fraction. Like I kind of just told you guys the system works like this, but like it's really hard to like really know it's doing that. So we really want to directly test it to see if this is really what's happening. Okay, so our idea is this. Fret and intensity are sort of independent measurements in some sense, right? Fret just comes from the shape. Intensity comes from the area under the curve. Unlike if I put a random sample on, there shouldn't be any correlation. However, this model predicts a correlation between the two measurements because of this molecular parameter and pol. If a position is brighter, it probably has more polymer, and it also has higher fraction. So in other words, they both depend on polymer in different ways. And if polymer really is this thing that if fret really depends on polymer how I say, I expect this relationship. So I'm going to test, when I make measurements of F and I, do I really see this relationship? So I'll take my sample. I measure I have an intensity here. So I take this xenopus egg extract. I add Taxol to form microtubules, which also was the drug at the very beginning I said is a chemotherapy. It's out of interesting stuff. OK, so I have this intensity. These are these large microtubule structures. I have this fret. Now I'm going to group pixels and measure the fret and intensity and see if I see this relationship. OK, I do see this relationship, right? This was like we were very, very happy when we saw this. I still remember when we first saw it. Because we built the system for a long time, did a bunch of tests, nothing was working. It was really hard to show it. And then we saw this, we're like, oh, sweet. OK, this is very likely good. But it's not the whole story, right? I still have two free parameters. And those aren't just arbitrary fitting parameters. So if the model is really good, I should be able to vary these experimental conditions and get these things to change as expected. That's what I'm going to do next. So the first thing I'm going to change is epsilon n mon, which is just the number of photons from monomer. So that number should scale with acquisition time. And you can think of acquisition time as kind of just exposure time, right? If I acquire for longer, I should get more area under that curve. So let's look at our thing. OK, the intensity is much. OK, so purple is like 50-second acquisition, and green is 10. And this is the same xenopus egg extract, adding Taxol, and then seeing these structures. So in the bottom part, it's really dark because the acquisition's low, and then it becomes bright when the acquisition's high. But the fret, the coloring more or less is the same. So we don't really see a change there. This is what we expect. The fret doesn't care about the brightness, the area under the curve. Both of the two samples fit really well. Now from this fit, what I'm going to do is I'm going to say, what's the best fit? And I want to make sure that this horizontal asymptote doesn't change, because that corresponds to this one parameter PF. But this x-intercept does change, which corresponds to epsilon n mon. So PF doesn't change at all. Epsilon n mon changes linearly as expected, so that's really good. OK, so this all checks out. Finally, we're going to check PF. So this one I think is a little more interesting, or I like it more. Here, the idea is if you're a donor, your probability of being engaged in fret is just linearly related to the amount of acceptor. And that's true in a low acceptor regime. So in other words, you're a donor, you only fret if an acceptor is nearby. If you double the amount of acceptors, you're going to double the chance an acceptor is nearby. OK, So in green, I have low acceptor, and in purple, I have high acceptor. You can see the intensities are pretty similar, but the fret goes way higher. And then higher fret is like a more yellow color. OK, so qualitatively, we got what we wanted. Let's go see quantitatively. Oh, it's really nice, right? This x-intercept is exactly where we want it to be, but this horizontal asymptote is larger, right? And again, on the y-axis is fret fraction, on the x-axis intensity. I'm gonna take this 2-parameter model, I'm gonna fit the data, I'm gonna say, spit out those best fit parameters, do they change as expected? Great, probability of fret did change linearly as expected. Epsilon Edmond shouldn't change because that just is photons from monomer, that doesn't change with acceptor concentration, and that's flat. Okay, so initially I was like, okay, that's all the parameters in the model, we're done, there's nothing else we can do. But there's actually secretly one other thing we can do, or possibly more, but at least one other thing we did do, which was actually suggested by Ethan, which is to do a microtubule dilution series. So just make some microtubules and dilute them, and make sure your measurement can track the fact you're diluting them. And so this kind of tests the last parameter or variable in the model, which is npol. And in this case, we're kind of tracking the sum of npol. So this test is done in buffer. So the other ones are done in extract, but this one we have to do in buffer. And if I do the dilution series correctly and there's no pipetting error, I expect a line. And so I'm pretty good with the line if you compare it to a nice established centrifugation plus. In blue is an established method, and it's consistent with that too. OK. So I'm just going to super summarize what we did. We wanted to make a new polymer measurement thing. We related the two things we measured. We saw that relationship. We were super happy. But then we said, oh, wait, we got 3 more parameters. They all changed as expected. And we were like, OK, good. We believe this thing works. If you guys are, so this general idea of trying to cross-correlating measurements to try to validate a spectroscopic one or both of them, I think could be applied to other spectroscopic measurements that are polymers. If you're interested, this is coming out this month in MBOC, and one reviewer called it well-written and pleasant to read. That's literally from the peer review. All right. All right. So let's go back to why we developed this whole polymer measurement thing. We did it because we want to see, do we have something short? And like the committee was saying, maybe it's not quite 1, maybe it's like 2 or 3, or do we have something long? So now let's go make this measurement. So first I should thank Olivia, who is a visiting scientist who helped take all these measurements. She was super awesome, helped debug stuff that would have taken like 1 trillion times longer if she weren't here. So I really like to thank her. OK, and so let's talk about what we're going to do. I think I need to go to the computer for this one. So first what I'm going to do is I'm going to take an intensity threshold, and from that get the spindle boundary, which is here in green. Depending on the threshold, depending on that value, I can make it a little bit in or out, and I can tell you how it affects stuff, the sensitivity in a bit. I'm going to look for all the points that are equidistant from that boundary, right? That's what we want. We want to see, as a function of distance from the boundary, how does polymer change? I'll start in the spindle, calling that negative distance, and then I'll move out of the spindle, calling that positive distance. In yellow are all the pixels who are, in this case, minus 5 microns from the spindle. And let's see as we move out. So what I'm going to show here is I'll show the fret fraction and intensity of these pixels, or the average fret and intensity of these pixels. And then from that, I can calculate the polymer concentration. And we're going to, you guys can look at whatever you feel like, but I advise looking at this polymer concentration curve. That's what I'm going to be talking about. OK. So what happens as we move? So here we're at minus 5. We move out. We move out. OK, polymer's been going up. OK, OK. Oh, OK. So now we're starting to go down. Now we're kind of leaving the spindle. Here's the soft boundary. OK, are we getting that long thing? Oh, it's pretty flat, right? Oh. Let's do that one more time just to make sure. So again, we're grouping pixels in yellow here. All of those pixels are grouped and give us that last point that's being drawn. I'll press play, maybe it'll be the easiest way. It's a little laggier than I thought it would be. OK, so this is just one spindle. It's a little bit noisy, but I don't really see any long length scale gradient, at least qualitatively. So what we're going to do is we'll do this on 3 separate days, 12 spindles, and see what we get. And we see I think basically the same thing, but with a lot cleaner data. So here, at least by eye, again, we see this really sharp drop. If I change the intensity threshold, these curves look nearly identical. All you're doing is moving this 0. Yes?
Does that change at all if you're actually just going along the sides of the spindles and ignoring the
Oh, That's a very good question. Okay, so I don't have the data here, but one thing we did was we broke up the spindle by angle to check out angle dependence. And they look all the same from different, at least by 4 quadrants, they look the same. And so we'll do more tests on that. But yeah, so like to see like, is it different? Because like in theory, you could be like, well, maybe it's just the like perpendicular distance to the chromosome that's important, or like the poles act differently. But at least from what we can tell by quadrants, they're all the same. Great question. Okay, so this all looks really good. So we're like, all right, we don't see any long length scale, but if you kind of remember, we were saying we think it should be 10 or possibly greater. So we're just going to check. Maybe there's some really long gradient that I just can't see because I'm so zoomed in. I think it's unlikely because this data is pretty clean, but just in case, we'll like zoom out and we'll see, okay, if I look at the 100 micron scale, again, it's flat. The spindle is basically just one data point here. Yes?
Going back to
Yes.
some of the earlier questions,
Yes.
It's not clear to me why you even needed to define a boundary. You had on this image just the threshold of where the boundary is, but really all you're looking at is the polymer concentration.
Yeah.
So why did you need to define a boundary?
Okay. Given this data, okay, so let me think. That's a good question. I guess what we were thinking a priori was, if it were the case that nucleators continue to, okay, so actually here's why I think we need to, that's a good question. Why do I have to even define the boundary? I know this, give me 1 second. Okay, I guess there's an implicit assumption that when you leave the boundary, you're no longer, if I measure polymer, it's diffusing polymer, not bound polymer. That's the assumption, that once you leave the bound, like I could imagine maybe you're out here, and like if microtubules like teed off each other all the way to here, if they were really bound to each other, that would be consistent with both models. So I'm kind of saying once you leave this boundary, any polymer I happen to see is from freely diffusing polymer.
But your polymer concentration on the right over here is both diffusing and
Oh, sorry. Yeah. Yes.
How would you draw that yellow line of choosing what, so here you have yelled out equidistant to what?
Yeah, so this is equidistant from the boundary. And the reason is, this is a really, okay. So now I remember. Thank you, Dan, thank you. Sorry, thank you. Okay. So the idea is we're turning on stuff in this, we're turning on nucleators in the spindle, right? So like first you turn them on at the DNA, but then there's this further cascade of stuff being turned on the spindle. We know if we don't see, so the idea is if we're turning it on the spindle, we're turning it on locally, but it's being turned off everywhere. So in other words, you have to turn off, what happens is you're turning off nucleators everywhere at some like low rate, but you're turning it on locally at one point. And so the idea is the boundary tells you at least what distance are you from the source of active nucleators. So I think that's the point of a boundary is if I had no boundary at all, like I have to center my measurements about something. Yeah, I totally left out. You turn on nucleators in the spindle, but then they're kind of turned off everywhere. And so that's why we have, that's my explanation. Okay, good. Yeah, sorry, I forgot to say that part.
So this boundary is then just defined by the intensity?
Yes,
right? So
it is.
How sensitive are your results to the...
Great question. Okay, so basically extremely insensitive. All it does is translate the 0. The shapes looks like nearly identical. I can't see the difference by eye for sure. So that's a good question. Okay, so we do a short, we do the long. Okay, we don't see anything. So I'm kind of arguing, qualitatively that it's not looking good for the all nucleation model. That's the one where it nucleates whether bound or not bound. But we can be a little more formal about it. All right, so if it's been a long talk for you, you want to take a little break, you can like sit back on this one. But I'll briefly go through what's being modeled in these equations. I need to thank Sebastian, who's this really awesome theorist at Simons, who previously NYU, and he's doing like this modeling and does like the PDE numerical solutions and all this stuff. Great guy. Okay, so the idea is basically we're saying that there are growing microtubules, and those microtubules can grow, they can diffuse, and they can turn to shrinking ones. Shrinking ones can be created by switching from growing to shrinking. They can diffuse and they can shrink. Nucleators, what a nucleation profile means is starting off a growing guy at a certain nucleation size, epsilon. The total density of microtubules is just the growing plus the shrinking. Nucleators can diffuse, and like I forgot to mention before, they get turned off at some rate RC. everywhere. And then finally, the thing that is like important, or whatever, it's all important, but the final thing I'm going to point to is TU is the activity of unbound nucleators, or tau U, and TB is the activity of bound nucleators. So this is where the two models differ. If you're a bound nucleator, if you're under the bound nucleation hypothesis, unbound nucleators can't nucleate, or in other words, TU is 0. Under the all nucleation hypothesis, you don't care about your localization to a microtubule or not, or you're binding to a microtubule, so TU equals TB. So most of these parameters we can grab from other people's measurements, like growth velocities, polymerization velocities, diffusion rates, all this stuff. I think we get like 1. I think we have to plug in an epsilon, and and then the diffusion coefficient of nucleators. You can pick your favorite nucleator, get a little bit 1. But basically, we think that the picture looks like this. So if we average 12 spindles, we get this black line here. And on the y-axis, we have polymer concentration. On the x-axis, we have distance. And then each gray line is an individual spindle. The average is in black. The bound nucleation hypothesis predicts this blue. The all nucleation hypothesis predicts this green. And you can see here, while we're pretty consistent with blue, green is not consistent at all. And we tried making green more consistent. And if you try to use reasonable values, you just can't make them. You can't make this green look like this data. So I like to think that we've ruled out the all nucleation model, but there's still a lot of work to do. So for one, we've ruled out the all nucleation model. It doesn't mean that unbound nucleators have no activity. Maybe it's low. You could imagine having a little bit of this green, adding a little bit of unbound nucleation is kind of like adding this green to this blue. You could have a little bit of that green in the blue and still kind of be consistent. So we're trying to get an upper bound on what we think is realistic for unbound nucleation. This is a very simple nucleation model. We can kind of investigate a little more complicated ones. And finally, we have these measurements of polymer, but from the same measurements, we get a monomer gradient as well. And so we're going to investigate monomer gradient mechanisms. Acknowledgements. All right. So first, I'd like to thank Autumn. So one of my favorite parts of the PhD was going to her science fair and getting involved with her students. So I really, really like to thank her. I also like to thank Will for carrying on our labs going. Where's Will? There's Will. All right. I also like to thank Will for carrying on our lab's work, or our whatever involvement in this. And so I just want to say, you guys should really judge the science fair. It was the most basically the most fun thing I did in grad school. Plus all the science was really fun. But it's half a day, and it's really awesome, and it's just a good day to get out of the lab. And the kids are really excited about it. And I remember some of your guys' projects. Yeah. I was gonna guess one. Yeah, I remember color blindness, right? Yeah, and then psychology, you look at the shapes. All right, nice. All right, good. All right, so it's right off the red line. So it's super easy to get to, and you get, it's coffee and it's fun and stuff. All right. All right, so then I'd like to thank my mom. So pictured here is my mom with me and my twin brother, who's also pictured here with my dad. I'd like to thank all of my housemates I lived with who had a profound impact on me, very positive. Here's Bryan Hassel, of course, Zain Ali, Jess, and my girlfriend Emily. Special shout out to Lucas, who came up from New York. Where's Lucas? Yeah, also excellent Luigi. All my drinking buddies, Will, Dan, Kelly, Zach, and Julia. My collaborators, of course, who made my last 6 months super duper fun. The whole Needleman lab, they've all been great. Maya and Frank really go out of their way to make the lab run smoothly, so I really appreciate it. Peter always helped me out. And finally, Dan, I just always really enjoy talking science with Dan. Actually, when I showed up at the lab, I'd never taken high school or college biology. And so Dan really helped me get up to speed. And he also taught me a lot of physics stuff too. So I always like talking with Dan. So thanks to Dan. NSF for funding. And my very last slide, since we have younger grad students and future grad students, I was just going to kind of run you through my experience, and other people have similar ones. So when you start off at grad school, you think you're the coolest in the world and that you're going to solve every problem, and you probably feel like Indiana Jones in this clip. So tell me if you guys can hear this. Hold on, sorry. Oh man, I knew this was going to happen. All right, this is just 10 seconds and then we're done. All right. Yeah, I'm far from year one. All right, let's try this again. Are you serious? Okay, last chance, and then it took me so long to get these like clipped and trimmed and all this stuff embedded. All right. Okay, let me try one more time before I try this voiceover. Let me just see. Oh, it's because the sound was low. Okay, now I think I got it. Oh my God, what's going on? Oh, there it is, okay.
It shock you?
Nothing shocks me. I'm a scientist.
All right. And that's how you are as year one. But then, 4 years of like failed experiments and things just aren't working and everything's so complicated and you just like, it's supposed to work, but it doesn't work. Then you feel like William Shatner cover of Rocket Man. Hold on.
Nothing.
And all this science, I don't understand. It's just a job. 5 days a week. Rocket Man.
Okay. And then finally, like, oh, sorry. Your stuff like eventually works and you have a lot of ways you can like use your measurements to study things and you really start to appreciate all the different styles of science and all the options you had to really study a problem or even think about a problem. And then finally, I think I see science the way master scientist Rick Sanchez does here. Let's see.
Okay, well, sometimes science is more art than science, Morty. A lot of people don't get that.
All right. Well, all right, and with that, I'd like to thank everyone. If anyone has questions, you just raise your hand. Question?
So, Bryan,
Yes.
I wanted to ask you
Yes.
about your modeling.
Yes.
Chapter 2.
Yes.
So, as I understood your model, right, it's a probabilistic model which accounts for
Yes.
photons that are arriving from the short decay and those from the long decay.
Yes.
But it seems like in that model you're assuming that every photon is counted. That is, you're not accounting for
yes.
measurement. So,
Okay, so one thing okay, this is a very good question. So it's fully, so there's an implicit assumption that the chance that you count a photon is independent of its lifetime. So if there was some reason that shorter lifetime photons had like a different angle, that it shot out, didn't hit, for whatever reason I didn't see it, yeah, that is not accounted for in the model. As far as I don't know any reason an emitted photon would, so it's true that guys who fret are less bright. So that is taken into account, but maybe that's the question. Is that part of the question?
I mean, there's different things you could think of. Like
Yeah.
you could have your detector have a false positive, probability, and
Mm-hmm.
a false negative probability, which you probably have both of in your
Yes.
detectors.
So what we have is, we do have a term for dark current and uniform background, so anything with an infinite lifetime is in the model.
But
Yes.
also just the blips you're getting, it could be a false positive event.
Yeah. That's true.
So if you account for those, it changes. I mean, even if they're the same for the two kinds of events,
Yes.
it would change your uncertainty.
It could change your uncertainty, definitely, yeah. It would probably make them less sensitive to changes in short lifetime or something, if I always had blips there or something like that. My guess would be.
Right. So I guess the question is,
Yes.
how much do you think that would affect the results?
So-
What you've shown in that.
Yeah, so that's a really good point. So, okay, when we do this FLIM modeling, there's a lot of assumptions, like things with different lifetimes don't have different stuff, and that we've really captured all the calibration issues. I mean, there's also a lot of other details I didn't go into. And I think that's one way we want to validate the polymer measurement further along. So it's always great to do your best on the FLIM analysis. And we have done a lot of sensitivity saying, oh, what if the lifetime were different than I thought, or if there was a third lifetime? And it doesn't seem too sensitive, but I think Yeah, so one thing to do is to see if your changes in fret fraction, that seems to be relatively insensitive. Changes in fret fraction is much more insensitive to perturbations than the absolute value. But I'd say that whole reason is why I think it's so important to sort of validate the measurement sort of at the measurement level. Because once I say, here's the fret fraction, I start plotting it versus intensity, that's fret fraction measured from flim. So whatever error there are in assumptions are kind of in that. And from what we can tell in the pictures, none of that seems to propagate to anything we can see. But it's a really good point. It's kind of one of the reasons I think that kind of elucidates one of the reasons we want to really validate the measurement. Yeah. Was there a question? Oh, question.
I had another question
yes.
Also in regards to sort of, you're doing two different measurements, one based on intensity and one based on the lifetime.
Yes.
But one of the things that's a little hard to deconvolute is there actually a difference in the fluorescence? A, of the fluorescence of your fluorophores as they get buried versus mono.
Yes.
B, also quenching.
Yes.
Especially within where you have so many sitting right next to each other. I expect that to be a lot more problematic.
Yes. Okay. So maybe I can show it. Okay. So I don't. So those are all very good questions. Let me address those. So, okay. So in a very simple system where there isn't anything too complicated, but let's just say we have fret versus no fret. The quenching is given by this alpha here, this relative brightness. And at least in the theory, the relative brightness is given by the ratio of lifetimes. So just give an intuition of why that should be. If I have two pathways to decay, this is a longer lifetime, and more of these guys are going down this pathway of emitting photons. When I add fret, I have another way of decaying, so less are going down this pathway, and also the lifetime is shorter. So these two things are linked, and that link happens to be that the ratio of the lifetimes, it gives you the ratio of the brightness. What that doesn't answer quite yet, which is a good question you asked, is what about being in a microtubule? Like, it's a complex environment. So I didn't show it here, but one thing that we did do is we formed microtubules without acceptor, and we saw the lifetime of that donor did not change. So that since the lifetime didn't change, it says at least in the absence of acceptors, there's no funny business between microtubules affecting, local concentration of endogenous quenchers and things like that.
Scaling the amount of the relative proportion of donors or acceptors.
Yes. So if we scale donors, we don't. So it's a good question. In the regimes I use this stuff, we scale stuff, and everything goes as expected. So I can change donor concentrations and everything looks the same. I change acceptor concentrations and it changes as expected. If you add if you add a lot of donor and acceptor, you can start getting weird stuff because I think what's happening is they're aggregating in solution or something weird like that. I'm not sure, but if you go to really high, I think you might start losing these assumptions. But
what percentage labels?
So we do, in all the measurements you saw, those were, and all the measurements of the spindles at least, because in the other one I like change stuff. It's 2 micromolar acceptor, and I think I wrote it in the thesis, but I think it's 2. Give me one. I'm pretty sure, and when you add both donor and acceptor, you're at 3 micromolar, and the endogenous tubulin's at about 20. So right around 10%, 10-15%. So that could be a little high. There could be some issues with the.
So what fraction,
Yeah.
Given the microtubule lattice,
Yes.
what's the probability of the donor being next to a donor versus a donor being next to an acceptor?
Yes, great question. Okay, so one way we can get at that is we can say, how does PF, which we get from these fits, okay, I know so I know or I have an estimate of the endogenous tubulin. I know how much I add, so I know what fraction are acceptor. Then I can say when I measure this probability of fret, which is given here, right, from these asymptotes, I can say how does this slope look with acceptor concentration? And when you look at that, you get a slope of 2. I'm trying to go back. So slope of 2, which naive interpretation, like a oversimplified one, would be that a donor can fret with up to 2 neighbors. If you have a slope, so it's like, do you see what I'm? Yeah, that's kind of a little oversimplification because this is all averaging over a bunch of stuff. So that might be the case, but it might be something a little more complicated. Yeah. Go for it.
Yeah, who are your top 3 favorite?
I don't want to answer this question. Thanks so. This is my brother. I love him, but yeah. Can't be on the spot like that.
All right, so no other questions?
Great job, Bryan.
Thanks, man.